introduced myself. I introduced myself. Yeah.
Just two lines. Associated
Dr. Surya Narayanan sir is a distinguished researcher in environmental monitoring and assessment with expertise in environmental science, water and wastewater treatment and impact assessment. He holds a PhD in environmental science from Bharatiya University along with an MPhil and MSc in zoology. His career reflects a wealth of leadership and international experience, having served as Deputy Director of Research at Vinayaka Mission's Research Foundation, Science Officer for Scope in Beijing, and Postdoctoral Fellow at the University of Turin, Italy. A recipient of the DST SERB Young Scientist Award and Visiting Scientist Fellow of the Chinese Academy of Sciences. He has contributed extensively to environmental research through his publications and collaborations worldwide. It is an honor to invite him to take the stage and introduce our esteemed speaker. Please join me in welcoming Dr. S. Surya Narayanan sir. Welcome you, sir.
Good morning, everyone. Seeking the blessings of His Holiness Swamiji, it is my distinct privilege to welcome you all for this today's session that sits at the fascinating intersection of neurosciences and metabolic research. Before we begin, I would like to request all the participants be seated. Please take your seat and request all of you keep your mobile phone silent for smooth conduct of this session without any interruption and active interaction. So the subject we are going to explore today, the lipid biomarker analysis in obesogenic conditions and human brain neurodegenerative diseases, represents one of the most compelling areas of contemporary biomedical research. Because we are all aware of that, the global burden of both the metabolic disorders of obesity-related problems and neurodegenerative diseases continues to rise at an alarming state. According to the recent epidemiological data, globally 650 million people are classified as obese, and also 55 million people are categorized into the dementia problem globally. So understanding the biochemical bridges between these two metabolic disorders and neurodegenerative diseases, this could be a revolutionized approach to for the preventive medicine and the patient care. It is my great honor and privilege to introduce our distinguished speaker for today's session, Professor Bommegowda Siddhe Basave Gowda. Professor Gowda serves as Associate Professor at the Faculty of Health Sciences, Hokkaido University, Japan, where he affiliated with the Department of Health Sciences and Technology. He holds a Doctor of Life Sciences degree and brings a unique interdisciplinary perspective that bridges synthetic organic chemistry with cutting-edge lipidomics research. Dr. S.B. Gowda holds an MSc in Chemistry 1985 and PhD in 1993 from University of Mysore with an academic career spanning over 3 decades. He has served as a lecturer at St. Philomena College, Yuvaraja College, University of Mysore and Government First Grade College, Kusal Nagar, and as an associate professor and head of the Department of Chemistry at Maharani Science College for Women, Mysore. As a synthetic organic chemist with diverse expertise in sphingolipid synthesis, analysis and biochemistry, Professor S.B. Gowda has established himself at the forefront of lipid biomarker discovery. His research portfolio encompasses 3 major disciplines: lipid synthesis and biochemistry, synthetic organic chemistry, and metabolomics and analytical chemistry. His current research areas of interest of lipid profiling for disease biomarker discovery using advanced lipid chromatography, mass spectrometric techniques. At Hokkaido University, he contributes to both undergraduate teaching in medical technology and postgraduate teaching in health sciences degree programs. His work exemplifies the bench-to-bedside approach essential for advancing our understanding of lipid metabolism in human health and diseases. Please join me in extending a warm welcome to Professor B.G. Siddhe Basave Gowda. Over to you, sir.
1 minute. Is not submitted. We have this one, no? Okay. Done? Oh, this one. Okay, thank you for the nice introduction. So, it's okay? Maybe. Should be fine. Okay, myself, Dr. Gowda from Hokkaido University. Probably, the professor introduced very nicely, but I'm still 39 years old. I'm not, probably the AI made a mess when they search about my profile. So AI is always not trustable. Like our Prime Minister says, when you ask an AI to write in, right-handed, it shows you left-handed. Opposite results. Don't trust AI always to. Our brain is most, we need to reinvestigate, think and rethink. There are some errors, but it's still fine, sir. No problem. I am not, yeah. Like as I said, I worked in Maharanis and all. No, I graduated from University of Mysore and then moved to Japan for my PhD, and then continuing there for last 14 years my career in Japan. Well, today because of lack of time, I hope I'm audible, right? Sleepy time? No? You got some energy, right? Had coffee or tea, some stimulants went to your brain now probably. It's very quick, right? Anyhow, today, because the theme of the topic is related to food science, right? Connecting the bridging chemistry and food science. I put two topics. Originally, I suggested this title, but I'm going to talk a little bit also on the food side and hopefully can complete within the provided time limit. Okay, I'm audible, right? If you have any questions, you can ask me anytime. Okay, let's move on. How to switch this? Should I do? This won't work. So next slide, maybe. It's not working, sir, actually. Have you connected this, right?
Yes,
connected. Maybe it's too far too. Oh, it's working.
It's working, sir.
Oh, okay. Well, Hokkaido University is like this, looks like this. Quickly, I can show you. Oh, okay. So we have that's the old Japan. So we are in the extreme north, where the Sapporo campus is. Sapporo is located. In that Sapporo city, we have this campus. Not big as JSSTU or Mysore University, but this is quite largest campus area even in Japan. And I'm affiliated with the health science department and the global food resource, kind of connections between the food and health, our research very close. So one is agriculture and one other one is a medical school. So both are within the campus. I'm affiliated with both the departments, and that's our train station, very close, like a 5 minutes walk to the campus. We're quite green campus, and we are #1 for SDGs in Japan. Well, my lab, I always choose probably with a couple of JSS students, will focus on these three keywords. One is on the lipid chemistry, because I'm a chemist originally and converted into a life scientist, and finally working in the area of food and health. Lipid chemistry, where we work on the synthesis of the novel lipids, which is challenging and which is difficult to get so far in any commercial suppliers, and also to isolate some bioactive molecules from, especially focusing on the lipids. And the second area we investigate is the omics, lipidomics, where we perform the analysis of the lipids, either whether it's in the foodomics, where we looking into the food composition, or we also go for the clinical lipidomics, where we look at the patients or the urine samples or the cell samples and look into the what kind of biomarkers we can detect for the early diagnosis or for to find the target for a disease. Such kind of studies we investigate. Along with that, recently some of the students started working on the functional foods to find the solution for obesity through food. So trying to make a molecule that can able to catalyze, control the fatty liver disease. So we are identifying certain molecules from that food. Well, with this brief, I will talk about lipids. as you know that lipids are the nonpolar molecules, right? Like whenever you mix oil and water, it never mix, always floating, right? So they are mostly nonpolar and amphipathic molecules. They're having a polar head and nonpolar tail, right? So like this, it looks like this. So it's like not working anyhow. So it looks like a long tail and the head molecules. So far, you know what in the world is going on in the lipids. Where the lipids are present? Everywhere in your body, everywhere in the food. Okay, the two ways we can get the lipids is they are the biomolecules, of course. They are essential for our body. One way is you have to produce in the body, right, for the cellular function. Another way we must take through the food. So all the lipids, we can't make it. So you need to take oils, such as omega-6 fatty acids, omega-3 fatty acids, linoleic acid. These things we must take through the diet. So far, you know, lipid science is still slowly progressing, not like a proteomics or other areas like genomics. But the lipid discovery or hunting for the lipids is still going on all over the globe, like a running race. Every year, the number of lipid discovery is increasing. And so far, we have reached about 50,000 new lipid molecules reported in the database, including some computationally generated and experimentally obtained. So every year, this number keeps increasing. And we do have like 5 different types of lipid classic categories depending upon their structure and functions. Like, you know, how do you group? You are Indian or you are Japanese or based on your, like similarly, here we make the group based on their molecular structure and functions. Well, today's class topic, I told you briefly, I will explain this to and end up my talk not in going very detail what we are doing it, just to give a hint. We are working on multiple projects. One of the projects was on the neurodegenerative diseases. So how, as I told you, lipids are everywhere, right? Everywhere in our body. So if something happened, what happened? This balance gets changed. Under normal conditions, everything is normal. Some enzymes take some substrate, make some product. It's in the normal conditions. But whenever there is an imbalance, what happens? Something go up, something go down. So the changes happened. So these changes can be tracked down as a markers. If you detect that, that's the same thing. If you're testing for a diabetes, you have to measure the sugar levels, whether sugar going up or down. Something like that, the lipids contributing to the disease, either in a positive way or in a negative way. If you monitor these levels, we can diagnose the disease or we can find a target of the disease where the drug need to act in, right? For that purpose, I show you a few examples with that neurodegenerative diseases. And second one is I put it in because it's a food conference. We are also one of the students is here, he works on seafood raw. What is raw? Anybody heard? I don't think I'm talking raw in JSSTU. It's a fish egg, or the crab eggs. So everyone knows chicken eggs, but this is from the fish eggs. So I will talk to you a little bit on that. Well, as neurodegenerative diseases, there are various types of neurodegeneration. What all these neurodegeneration mean? Loss of neuron function. We have several neurons, right? So these neurons get degenerated. They don't function normally. Leads to the neurodegeneration, such as Alzheimer's, Parkinson, Huntington disease, and sclerosis. Several others, each having a different, type of neuron dysfunctions. I don't go in detail about each, but so far these disease cases keep increasing all over the globe, and there is no cure for these diseases. We don't have any drugs to stop this disease. Another challenge is how to track, how to identify whether I'm getting a dementia or Alzheimer's. This is also challenging. So one way we can find out diagnose the disease is MRI imaging. Whenever you scan your brain for MRI and see what happened in the brain. So that way, once you got the disease, we can identify. For example, the left one is a normal brain. The center one is called MCI, mild cognitive impairment, where the gray, white things started disappearing. Can you see the black holes a little bit? So that is much severe in the AD, Alzheimer's disease. That means gradual loss of this gray matter causing the disease. We can see in the MRI images says that this patient having this disease. This is a doctor's case. But how we can detect this earlier even? Well, a routine blood check, routine saliva test, or something else that can be tracked down early before getting into the disease. So there are several protein markers so far, but since our area is mostly on the lipids, we all know A beta accumulation, everything. There are lots of mechanisms, but this accumulation of proteins always connected with the lipids. Lipid-protein interaction is always important. That's why many people work on the multi-omics approaches. Look into the gene, look into the lipids. All you can integrate the data and find the target. What is the target? Well, one of the study we conducted is, this is a human brain sample analysis. Quickly I move into the results. Containing the few patients from the healthy brain. That means that nobody will give the brain anyhow. This is the postmortem samples. Once the patient is dead, we can collect the brain tissues and look into what is the healthy brain, healthy person brain is. What is the AD brain is. What is the HD, Huntington's and Parkinson's. And we take the different sections of these brains and look into what happened in the brain, why the disease occurred. So this was a cooperation study we conducted, and all these patients from the samples from the United States. So this is the workflow we do. I don't talk too much, just get the lipids from that, extract it, send it to a mass spec, LC-MS, LC do the separation, mass do the detection, get the spec data done, and visualize what happened. Looks very simple. Takes several months to run it. Anyhow, we did that. So what you can see here, very simply understand that, as you can see, left the red one and the green one, right? Green one is the healthy volunteer's brain, and the left one is the Alzheimer's brain. They are elliptical and grouped together. That indicates, the profiles are different. The healthy brain and the Alzheimer's brain, lipid content is changed. These diagrams, all the cases, all the neurodeals. What kind of lipids causing this separation? This kind of some certain lipids, which are listed over there, down, may contribute to those lipids may be changed in between this. What exactly we found, AD, PD, SD, just a simple one. Alzheimer's, we found this as a marker. So what happened, this is a lipid called phosphatidylserine. This is an amino acid and this is a phospholipid. So this is mostly present in the healthy brain in a larger quantity. I mean to say, good, without any oxidation. But what we observed, it get oxidized because it has a double bond here. Oxygen can go and attack and get hydroxylated, and getting a oxidized phosphatidylserine. As you can see, the AD near, the pink color one going up very much compared to the healthy brain, right? Very much increase. Can you see the 5 to almost 25? 5 times accumulation. That means those molecules getting accumulated. Why it is accumulating? We don't know. What is the triggering factor? That's another thing. We have to investigate the mechanisms. But this is, we are just a descriptive results, what happened in the brain. That's in the AD. Whereas PD, as you can see here, some of the lipids like ether type, this is again phosphatidylserine, but we're having an ether connection and the ester connection. Such lipids goes up, increasing, deposited. Whereas PI, inositols, like this kind of lipids, decrease drastically. This is interesting because PI is very, very important for our brain. The phosphatidylserine makes your neuron functions. So they go down, mean something happened in these patients. Okay, we identify which region of the brain as causing this, mostly occipital region, the one which we call it as O, where the red you can see, those accumulation of those lipids. Those lipids are, where the blue is there, that strong blue indicates the loss of those lipids. Decrease and increase, just imagine like that. So most probably PIs were down in the occipital region, only one region, not all the other regions are impacted. Only particular region of the brain causing this. Now if you know the particular region, next thing is, what kind of particular cells present in that region? If you know the cells, we can now next go to the, revealing the mechanism. What kind of cells, whether it's a kind of immune cells or with other cells. So we can reveal its mechanism and identify the drug for treating these diseases. Well, these are the things. We have checked various regions. Addition to that, those PS and PI lipids, we also found some, especially for Alzheimer's, only Alzheimer's. We can see these are the patient's brain samples, healthy versus the AD, have a very high levels of accumulation of this lipid. Mostly, many lipids get accumulating compared to the normal brain. So these are the deposited and the depleted. Those who lost are here. Those who are increased are here. Just simply. With that, I can conclude this with one more slide that inositals are the key important for making a PIPs. PIPs are involved in the cellular signaling or the neural functions that goes normal. This is known thing. We know this. PIPs, PIs are very important. They are going down, leading to cause of this disease. And sphingomyelin, actually, only 1 or 2 papers says about this. That too, even from the Japan, says that the sphingomyelin accumulation is a key link to the Alzheimer's A-beta accumulation. So we found this sphingomyelin has a good correlation with the A-beta accumulation. So that indicates, if you measure sphingomyelin levels early, of course, this is a brain, but we can look into the other things, where the sphingomyelin is going into the brain, how it is going. So such kind of conclusions we draw it. So these are the things. And all this study was conducted by our JSSTU student, Jay Shankar. Maybe he's graduated as a PhD student. So that's I would like to acknowledge him, right? Well, secondly, because of the time, I will just quickly take another few minutes and then conclude. So second work, as I told you, we work on the foodomics. This is completely different, right? Biomarker versus foodomics. What is this? It's a fish egg. So mostly fish eggs were not consumed. They're discarded as a garbage. But in Japan, some of the selected fish eggs were taken as a food because they're rich in omega-3 fatty acids, good for your brain, something like that. Okay, they look like that. Well, what is the thing is, there are some studies says that if you feed my egg, PC or the phosphatidylcholine, like a normal chicken egg, it can cause the fatty liver. Liver gets more fat accumulation. But if you feed the fish egg, your liver becomes very normal. Imagine the opposite functions. What is the thing? What are the content in the fish egg responsible? It makes your liver healthy. So I asked the student, maybe Somitani is here somewhere, my master's course student. He's worked for 2 years with me. So he did this study. I collected all the eggs available in the market, asking directly from the fish companies or something, and we did a profiling. Which fish egg has this essential biomolecules that could help the liver function or the brain function? So we collected different fish, not only limited to fish, we also did in the octopus, crabs, eels, different varieties. Well, we found some differences. We identified about more than 400 lipids. What is important is here, as you can see, most of these lipids, the blue color indicates glycerolipids, like a triglycerides, are very much in this egg. Glycerolipids, like a TG we call it as, right? Triglycerols are very much in this species. Salmon or whatever we are eating. Salmon mostly widely used, these are the most. Whereas certain types of lipids, especially squid and octopus, not making the TGs. It making glycerophospholipid, PC. I mean, the red circle, as you can see, is the very highest, right? The longest. That means the more percentage of PC type lipids were present in this. Quite different from the other types of eggs. Well, if you're looking at some of the monounsaturated fats, in the heel egg, we found mostly eels making only monounsaturated things. Mono mean only one double bond containing lipids were quite abundant in the heel egg. What happens in the salmon egg, salmon raw? As you can see, these are the glycerolipids, like a triacylglycerols, containing this 25 or 22,6, whatever the isomers you say, they are the EPA and DHA. So these two are the most important fatty acids for our human brain. Very much important. Of course, if you are a vegetarian, so you can take the alpha-linoleic acid from the flax seeds, whatever. But this alpha-linoleic acid further converted into DHA and EPA by certain enzymes in our body. But those who are taking directly fish or such a kind of things, they can have this. What important thing I need to highlight here is salmon raw. Only in the salmon, as you can see, these lipids are highest. not in the other varieties. Only in the salmon, TGs are abundant. TG containing EPA and DHAs are abundant. What happens in the squid? Just remember, salmon versus squid. Quite opposite. Squid egg shows that mostly those two, right? Diamond squid, whatever the squid which we are seeing is, they contain the phosphatidylcholine, PC type of DHA and EPA are increased. One is giving a triglyceride type, another one is a phosphatidyl type. What is the function? So both have the good benefit. What is the benefit of TG DHA? It goes to the heart. It improves your muscle. There are some transporters. They can, once you eat these things, they go to the bloodstream and transport to the heart and makes you muscle. Muscle functions or liver functions can be improved. But those with the TZ, what happens to the phosphatidylcholines, PC type, those present in the squid, they go to the brain. See, one egg supporting you, making your liver healthy, another egg supporting you go to the brain. So if they are consuming only salmon, you are losing something. It is not reaching the brain, the molecules, because we found certain transporters that can transport these lipids into the brain. So we observed such kind of diverse results. This is just a profiling we are doing it, which fish egg. So next thing is what? I can't eat it raw. I can't eat egg. We can extract these things and process it and make it as a supplement. Take it as a 1:1 ratio, one with the PCDHA, one with the TGDHA. Mix them as a supplement, and if you consume them, probably your heart health and your brain health could be good. So this was the conclusions, like what we found in it. That's all. This is our collaborations. Lots of collaborators across the globe. One of the work, I think, are several, I don't want to mention some of the top works we published. For example, recently, again, JSS2 student was involved in this. Puneet got published in the Gut Journal, which is an impact factor of 25.8. So our lab doing a lot of high impact publications and in the top journals, even as a cover arts too. The students working hard and doing the things great. So these are some pictures with affiliate, JSS2 students visited our lab last year, and this year also some faculties are coming, and Dr. Maheshan sir was visited also last year. We're doing a lot of exchange activities with Mysore University and JSS2 and CFTRI. And this is my team, including several Japanese and Indian, Bangladesh, Chinese. So quite international. Now we have also from Africa. So quite international team we are running in. And those two students with arrows from the JSSTU worked with me for 3 years, and they got their PhDs recently. With this, I would like to conclude my talk. Thank you very much for your kind attention.
Thank you, Dr. Gordon. Before we move on for questions and interactions.
I took more time. Any questions? Sorry, I took more time. Go ahead.
Very wonderful lecture.
Yeah, sorry. Yeah.
In fact, we are also working on neurodegenerative diseases. They are curable, right?
Very difficult to say that at the moment. Yeah, probably if you find a potent drug, yes.
But we are looking through the gut-brain axis.
Oh, that's another one, yeah. That's another axis we need to think. Now, what we are seeing is the brain axis. We say gut is our second brain, whatever you eat. So that's also some people. We have that work also. I did not present it. We did the fecal profile of the AD patients, the feces, blood, and the saliva. This is now very soon, it will be out in some Nature journal. We submitted already this work and accepted. So we are also looking in this angle by collaborating with one of the collaborators in the United States. Actually, he's working on mostly into the metagenomic things and look into it. But yeah, we don't know whether bacteria will be the next medicines for this disease or any drug.
Fish waste you are using for what are the other purposes?
Which one?
Fish waste.
Fish waste, yeah.
We also use for omega-3 fatty acid extraction and a protein.
Okay, protein.
We use for some other purpose also?
There may be, I don't know, but mostly I heard this is discarded.
Yes.
Yeah, not so much consumption, like, yeah.
Wonderful lecture.
Yeah, thank you.
Thank you. And just I wanted to know whether this
Alpha-cycline
interaction
with DPA.
This is, we haven't checked that. But some lipids as sphingomyelin with A beta, we have some connections, we have stories. But alpha nucleosine, yeah, that protein, I think we haven't checked yet. Yeah, could be, yeah. Yeah, I told you, this lipid and proteins are always linked together because lipids are the metabolic signals. They from one protein to another protein, they're making some connections. True. Yeah, I totally agree. Yeah.
Thank you.
Yeah, thank you. Yeah.
I have one question. These lipid profiles, how it will be helpful for early diagnosis of
This is going on. Some of the tests, at the moment, still we are not into the hospitals. But probably after 30 years or 40 years, we can see such tests if they are validated, very long, very much. So there are some in Japan. Probably there are some lipid molecules which are identified as a markers. They are developing kind of a kit, diagnostic kit. Use that kit to check the lipid, and we can, like you do the blood sugar test or something, we can do them in our bloodstream. One way is to check the blood. But I don't think these tissue things, whatever we are using, liver or these things, just for revealing the mechanism, what happening in that site. But mostly through the blood, we have all the lipids carried by the lipoproteins. So we can find we can able to set up a method for the diagnosis, early diagnosis. It takes time, but this is a start. We can say we are just starting this kind of, not only now we studied the United States, but we need an Indian cohort. We don't know what happened in the Indian population, what happens in the European population. So that's why they are making a multi-cohort studies sometimes and validate the data, find a potential marker, and take it to the clinical level, like in the hospitals we can use them.
Your study clearly establishes the need of the collaborative research for this.
Yes, definitely. Yes, I agree, yeah.
With this, we are concluding this first session. We would like to thank, on behalf of the organizing committee, we would like to thank Dr. Gowda for the enlightening session on application of lipid profiling in neurodegenerative diseases, as well as very interesting to see the results on the lipid profiling of seafood roe, helping for the young person as well as the teleconsultant. Thank you, Dr. Gowda, for your wonderful presentation. We thank all the participants for your patient listening. Thank you all.
I thank Dr. Surya Narayan sir for chairing this session. Thank you, sir. Distinguished guests, respected speakers, and esteemed participants, I, Vidyashree, a second-year MSc Chemistry student, Department of Chemistry, JSS STU, take immense pleasure to be a part of this international conference. Now, we have second invited talk from Professor Taro Soni sir, Professor, Dean of the Graduate School of Global Food Resource Faculty of Agriculture, Hokkaido University, Japan. I request Professor Taro Soni sir to please take the seat on the dais. Now, I welcome Dr. N. Hariprasad Professor Sir for chairing this session. It is my great honor and privilege to introduce the chairperson for this invited session, Dr. N. Hariprasad Sir, respected Professor and Dean QAA, Department of Biotechnology at JSS Science and Technology University, Mysore. He has been a dedicated academician at the institution for over 2 decades, having served as professor from 2003 to 2022, assuming the position of dean. He holds a PhD from the University of Mysore, and his area of expertise lies in biotechnology and applied microbiology with a specialization in medical biotechnology. He has made significant contributions to research with multiple publications in reputed journals. His scholarly impact is reflected through numerous citations and a commendable Edge Index, highlighting his sustained contribution to the scientific community.
With his vast academic experience, leadership, and expertise in the field of biotechnology, we are truly privileged to have him chair this invited session. On behalf of the organizing committee, I respectfully invite Dr. N. Hariprasad sir to kindly take the chair and guide us through the proceedings of the session.
Very good afternoon to one and all. Indeed, this session will be very useful for all of us here. It's my proud privilege and pleasure to actually introduce and welcome Professor Tirosone, a distinguished academician and renowned scientist from Hokkaido University, Japan, who joins us today as our invited speaker. Welcome, sir. Professor Sone currently serves as a professor in the research facility of Agriculture Laboratory of Applied Molecular Biology at Hokkaido University. He also contributes as Director, Strategic Advisor to the Global Center for Food, Land and Water Resources at the university. Having obtained his doctoral degree in agriculture from Hokkaido University and has since built an illustrious career in applied microbiology, plant microbe interactions. His academic journey includes postdoctoral research at the University of British Columbia, Canada, and faculty appointments at several Japanese universities before he joined Hokkaido University as professor in 2017. Professor Sone's research focuses on applied molecular microbiology with emphasis on plant microbe interactions, molecular mechanisms of plant diseases, and microbial dynamics in agricultural systems. His pioneering work has deepened scientific understanding of pathogen resistance mechanisms, microbial ecology in crops, and microbial applications in sustainable food production. He has published extensively in leading international journals and has received several recognitions, which include Hokkaido Branch Encouragement Award of Japanese Society for Agricultural Chemistry for his outstanding contributions to the molecular genetics of plant pathogens. Beyond his laboratory and research endeavors, Professor Sone plays an active role in several scientific societies, including the Japanese Society for Agricultural Chemistry and Japanese Society of Plant Pathology, where he serves on editorial board and contributes to advancing agricultural science and innovation. Professor Sone's interdisciplinary engagements embody the spirit of today's conference theme, Bridging Chemistry and Food, an integrated approach for a sustainable society. It's a privilege. We are deeply honored to have Professor Tirosone with us today, and I now warmly invite him to deliver his presentation and share his valuable insights on need of interdisciplinary research and education and food resources in current global challenges. It will be a very enlightening one. Please, sir.
Okay, thank you. Okay. Thank you for the nice introduction. So, okay, maybe I should talk like this. Okay, yeah. We like to emphasize about the collaboration between the different disciplines. Okay, about the wine research. Okay, let's begin my talk. これじゃないのかな。大丈夫ですか。Which どれ ？Slide doesn't work. Looks like. あれああ What time lag? Okay. So first of all, I will introduce my university, Hokkaido University. We are celebrating 150 years of our university. It was started in 1876. At the time, it was started at the agricultural college. So, but after how many? Many years, our graduate school of Global Food Resources launched in 2017. So I should say that our graduate school is very new in the history of the Hokkaido University. We started in 2017 for a master course and 2019 a PhD course. Okay, so this is our building. And this graduate school of Global Food Resources, we must investigate the issues relating to the food, such as food resources, food waste, food safety, food environment, water quality, desertification, public affairs. So addressing the starvation and poverty are critical from a global viewpoint. So to solve these kind of issues in this century, we need to extensive knowledge, the commitment to seeking the solution. So as a strategy to respond to these issues related to food, land, and water resources, we have many issues. So to construct a unified technical system and the locus for the human resource development. So we started this graduate school of Global Food Resources. So in the education in this graduate school, one of the important keyword in the education system in our graduate school is global and local leaders. So we foster global leaders who can contribute to all regions from a global perspective. So we have a kind of system of the education. We call it T-shaped education. So we need, in the master's course, we have a very, very different lectures from the basic science to applied science, including the some of the the human science, and also for the social science and political science, and also we teaching production of the foods such as agriculture and food making. Okay, so and in the master course we have some active learning system. That is WANDERVOGEL on-site experimental education, right? So and in the PhD course, master course, we have 3 years to deepen their specialty. So in the master course, they have the lectures. They will get the lectures on all interdisciplinary studies. And also they have some experience of outside to visiting the other countries. And in the PhD course, they will deepen their specialized area. So that we call T-shaped the education system. So our education policy is most advanced technology in food production, world-class professors or for all lectures, integration of humanity and sciences, integrated knowledge of food, land, and water, and WANDERVOGEL on-site experiment education and active and problem-based learning. So it's very rare that all activities such as lectures, seminars, experiments, and field works are conducted in English in Japan. It's very rare in Japan. We are doing everything in English. So another thing in our graduate school is that we are inviting many lecturers from all over the world. So we are giving the lectures in these all different kinds of areas. And you may find Goda Sensei, Professor Goda here, right? He is taking care of the cuisine and health, and he's working together with the UC Davis. And we have now the Professor Siddharth Chandra from the University of Mysore in the lectures of our curriculum. So inside of the circle is Hokkaido University's staff, and outside the circle is the staffs from outside. So inviting the distinguished scientists to give the lectures for our graduate school. And I will introduce a little bit more about the WANDERVOGEL study. We have WANDERVOGEL-I and WANDERVOGEL-II. So this time we are visiting here as a WANDERVOGEL-II. The WANDERVOGEL study is in developed country. We are sending students to Denmark to learn about how they harmonize the food production, agriculture, and the environment. Because the Denmark is one of the countries in the EU where they have the very, very strict control of the nitrogen amount. Okay, so they visit many places about biogas power plant, swine farming, and also the water resources to learn how they're doing harmonize the such food production and environment. And the WANDERVOGEL-II is to realize the current issues in food resources in emerging economies. Okay, so we first designed this trip to visit Myanmar. For 3 years we went, but the situation in Myanmar is not good. So we changed the place of this WANDERVOGEL-II. So in Thailand in 2023, 2024, Vietnam and India. Okay, in this year we are coming. We are sending our students together with the students in here, making a team to send some places to learn about the current situation, current issues in food resources in the area. So I'm very excited to what will happen to our students after this WANDERVOGEL study. So this graduate school have the career path. It's a very rich in variety. The international company, Japanese company, and government officer or national institute or some PhD granted ones will take the place in the university. Okay, so let's go to the next topic. So the next topic is in the graduate field of global food resources, influence research activities of faculty members. So when we started this graduate school, I have no collaborators in the different faculties in the Hokkaido University. But after we started GFR, we are doing the research about wine production in Hokkaido. Okay, so the wine production needs very large area of the research. Starting from the environment, and if you want to make the wine, you need to grow grape. So we need a viticulture, and after we harvest the grape, we need a fermentation, and we need to sell to the consumer. So that's large area of activity needs many different kinds of research. So during the wine study in this graduate school, we are giving the wine lecture to the students, and sometimes students will visit winery to harvest, and also we have a lecture about tasting. Okay, so students also will get the very wide range of the studies. during this study, this research, I have collaborated with one of the faculty member, Professor Kawaguchi, who is who are doing the research about the chitosan, showing in the upper right. That is the molecule of chitosan. He changed the molecular weight of the chitosan into small one, and used for the grape cuttings. So after cutting the grape tree, we can put into the soil to regenerate the new plant. But if we do that using the chitosan in very, very small amount, but you may see the difference of the growth. Non-chitosan, it's not so grow so much, but with chitosan, it's growing very, very much in June and also October. Okay. And next one is this is my research. That's about microbiology. We get an endophytic bacteria from grape. That was actually bacillus, and it has an activity to fight against some pathogen in the grapes, such as Botrytis cinerea, that is a gray mold, and Agrobacterium vitis, that's a crown gall. So if we put together both of the bacillus and the pathogen, the gray mold and the crown gall was reduced. Right, so such microbiology is also for working in the wine making research. And also we are doing the research about the aroma compounds in the wines produced from the Hokkaido local grape variety Yamasachi. So we need some chemistry, right, to extract the aroma compounds, and they know about the difference in the concentration in each wines produced from Yamasachi and other 2 red grape varieties. We found these 7 compounds, aroma compounds, which is rich in Yamasachi variety. And also, sorry, I put the words too high, but this is a lipidomic analysis or metabolomic analysis, which was done by Professor Gauda about the Yamasachi and Tsubaga and Kiyoma. These 3 are red grape variety in Hokkaido. So he extracted the compounds from these 3 different grape varieties and did the metabolomic analysis, and he found that in Yamasachi, Yamasachi is rich in vitamin E, and in Zweigert, it's rich in tryptophan, that is aroma precursor, and in Kiyomai, it's rich in aroma antioxidant in exiform. Okay, so we're working. So Professor Kawaguchi and me and Professor Goda and other more professors in the GFR are working together in this wine research. Okay, so this is the last slide I have. So I give you about the information about our graduate school, and also some examples of the working together in different disciplines. So that will make the, how can I say, as a larger perspective about how to make good wines, how to promote the wine industry in the region. But that's such a collaboration, including the other, the global universities, teachers. So that's in the meaning of global, right? So global is think globally, act locally. So graduate food research is inevitable for the sustainable future. Interacting with researchers, people from different backgrounds allows us to make global. Thank you very much.
Thank you, Professor Sone. It was really wonderful. You brought out not just how important collaboration is. Your university is a very good example of not just historical development, but historical development of collaborations. You call yourself the young university in your history of universities. That's very true, but it has grown with all the collaborations together. Taking one example of wine production, you very well demonstrated how everything across science is very essential and collaboration is the key to a successful outcome of research. Thank you very much. The floor is open for questions now. Come on, students. Yeah, I will. But students, he is the key. So it's better you interact with him.
Dr. Kane, very nice presentation. My question is like, what is the role of the low molecular weight fighters on the interaction for the break production? Do you have any interaction or kind of a mechanism to analyze using the low molecular weight fighters?
Aromatic precursor.
Yeah.
Yeah. That's aromatic. We actually, I should say that we have started just a few years ago. So we in the aromatic research we just started, and we found some the compounds in these a few years. So we have not started with the so much interaction with the other, producers We still we need more research about to give such kind of information to the producers.
Are you working as a plant growth promoter, in other words, kind of such plant growth promoter kind of?
So the yes, the bacillus work. Yeah, it's working as a antagonist to the pathogens, but also such kind of bacteria may work as a plant growth promoter, and also the chitosan, right? Chitosan is chitosan itself is known for many years, but that way the way of the known is the to put the larger molecular weight chitosan to the field, and we need to wait several years. But Professor Kawaguchi's work is he made the molecular weight to smaller one, and that it worked immediately after putting. So we are now thinking how to make it possible to use in the real field. Yes. Thank you very much.
Any more questions from students? His topic was very interesting, multidisciplinary research and collaboration, and very well executed.
Okay.
Anything? Nobody wants to go to Japan. Okay. That's good. He's always available for you to contact and interact with him.
Yeah.
In these few days he's spending here, and you can contact him also through your online media. So yes, sir, it was very enlightening. You have very well exhibited that interdisciplinary research is the key, and it is not just something you rightly coined the word local, wherein that is the answer for future. Because unless and until it is very true that a human being is first local, then collaborates with interdisciplinarity to become global. So rightly coined, and that is the impact of today's research, which every one of us should embrace. And very well, I should say, your university has very well amalgamated with the WANDERVOGEL program, very nice program you are organizing, and very interesting for students. Students should really enlighten themselves and utilize this. So I finally thank Professor Sone for his wonderful enlightening lecture, and also I thank the organizers for giving me this opportunity to chair this session, and also all of you for having listened to Some's lecture and enlightened yourselves. Thank you.
Thank you very much.
I thank Dr. N. Prasad sir for chairing this session. Thank you, sir.
Click the first.
Thank you, sir.
Now we have an invited talk by Dr. Prakash M. Alami sir, Microbiology and Fermentation Technology Department, CSIR Central Food Technology Research Institute, Mysuru, India. I request you to kindly take a seat on the dais. It's my immense pleasure to introduce Dr. Sadashivamurthy PM Sir, Professor and Head of the Department of Environmental Engineering at Sri Jaichamarajendra College of Engineering, JC Science and Technology University, Mysuru. I welcome you, sir. Dr. Murthy sir is a distinguished authority in environmental engineering with expertise in water supply and sanitation, air quality modeling, solid waste management, and environmental impact assessment. He holds a PhD in civil engineering sciences and has contributed significantly to environmental education and research. He is a fellow of the Institution of Engineers and serves as a member of the expert subcommittee on circular economy under CPHEEO, Ministry of Housing and Urban Affairs, Government of India. He is also actively associated with several professional bodies, including the Institution of Values and the Rivers of the World Foundation. Dr. Murthy sir has played a key role in major consultancy projects and international collaborations, and under his leadership, his department has honored with the prestigious Indira Priyadarshini Environment Award. We are privileged to have it with us today, and we will now be introducing distinguished speaker, Dr. Prakash M. Halami sir. Please join me in warmly welcoming Dr. Sadashivamurthy sir. Welcome, sir.
Thank you. Thanks for the nice introduction. Good afternoon, everybody. Already we are 45 minutes late. I had just spoken to the speaker before introducing him. It's my great privilege to introduce Dr. Prakash M. Halami, an eminent scientist and distinguished academic leader with over 3 decades of exemplary contributions to microbiology, fermentation technology, and food science. Dr. Halami currently serves as Chief Scientist and Head of the Department of Microbiology and Fermentation Technology at CSIR, Central Food Technological Research Institute, CFTR, Mysuru, and is also a professor at ACSIR. He has additionally held the responsibility as Head of the Department of Food Protectants and Infestation Control from 2021 to 2024 and served as the Nodal Officer for CFTRI COVID-19 Testing Center during the pandemic, significantly contributing to public health initiatives. Under his leadership, the MFT department was recognized as the best R&D department for the year 2023-24 by CFTRI. His research expertise spans genomics of probiotic bacteria, antimicrobial resistance in the food chain, and gut microbiota studies. Dr. Halami has successfully guided 16 doctoral scholars with several more currently pursuing research under his mentorship. He has authored 146 publications in reputed international peer-reviewed journals and holds 4 granted Indian patents. With over 200 conference presentations to his credit, He has received 27 Best Paper Awards in recognition of his impactful research contributions. In recognition of his scientific excellence, Dr. Alami has been elected Fellow of several prestigious academics and professional bodies, reflecting his outstanding contributions to microbiological sciences and food technology. With his vast experience in research, innovation, mentorship, and scientific leadership, Dr. Prakash M. Halami continues to inspire the scientific community and contribute significantly to the advancement of food safety, microbiology, and sustainable technologies. It is indeed an honor to have such a distinguished personality with us today. Dr. Alami, the floor is yours.
After two speakers from I will take a little bit support of this one. After two speakers from Japan, now you have speaker from the local institute only. In fact, before receiving PhD from Mysore University, I had a postgraduate training in Japan. It so happened all Japanese guests actually left, so I can't talk whatever the knowledge I had of Japanese language. That was actually JICA training, and since no Japanese delegates are here, so I can't say anything. At the outset, I thank the organizers, Dr. Mallu, specifically for inviting me to give this lecture in this wonderful program on international conference, bridging bridging chemistry and food. And you know that I am from the Food Technology Institute, and that is actually the right things to talk, especially during a lunch break, right? And as such, Mysore is known for a Food Technology Institute because one side, a very neighbor to you people is a CFTRI, and of course we have another institute is the DFR. Now it has been renamed as a DIBT, and then several actually institutes. You have a food science, food technology, food science and nutrition courses. Thank you, Chairman sir, for introducing, and of course our colleague is there, Dr. Sanjay, who is from the Biotechnology Department, and our great student is also there, Shreyas here. I believe he is a faculty. Chemistry, of course, is a little bit a different subject in a food technology or in food science. We have very close subject is a biochemistry, right? But then if you see, the origin of life or the food as such is a chemistry, Food approximate is nothing but the chemistry. Food contaminants detection is the chemistry. Profiling is the chemistry. So food actually is a chemistry, which we talk less, in other words. But then these are actually the subject goes parallelly, hand by hand, unless and otherwise you know this chemistry or the food chemistry, in other words, understanding the biochemical or chemical composition of the food becomes difficult, And now because of this FSSI, which I am associated with, that BIS regulations, the chemical approach or chemical aspect or profiling, contaminant detection, the regulation actually is just so important. I don't have many slides, of course. 20. Chairman sir told how much time I will be taking. I will not take very long also. 20 slides only. So 20 minutes you can say. Okay. CFTR actually has now 75 years. You people have seen the Hokkaido University's history from 14th or 15th century. We are not that old, just a 75-year-old. And it so happened, 28 years back, I was in Japan for the postgraduate training. So you can actually make out what may be my age now, right? And then of course in 2010, again in the JSPS program, I was there exactly in the Hakodate, which again comes under Hokkaido University, okay? So in this next 20 minutes, I will be talking with reference to the microbial metabolites, since I am from the microbiology department, and we are actually looking for a developing or using the microorganisms as a tool to formulate new functional food, right? A novel food. So how microorganism, especially the grass status microorganism, being used these days for formulation or a production of a different food products which are useful for our health and wellness, yes, right? And then you know that several of this enzyme which comes, the fermented product comes, are actually the product of the biotransfer molecules which are more useful than your the raw material, right? And then one such product you will see, as I told already that Japanese, most of this delegate has gone, the Japanese fermented soya bean, you know, is actually very rich in the antioxidants, rich in several bioactive molecules and considered as a functional food. So Foshu, the term which actually being used for the functional food, comes from the Japan, you know. So such few products which I will be describing. Here just we have actually the information pertaining to the CSIR, which is again the 84 years, you know, and I hope most of you people are aware that all this, our institute's chain of 37 laboratory comes under the CSIR umbrella, which is of course celebrating the completed 84 years in last September, okay. Yes. You see actually with the food system production, we know that we are actually self-sufficient in food production, right? But the growing populations, the malnutrition, the food security problem is concerned, the hunger nutrition, you know, these are the actually new challenges, the increasing burdens of the lifestyle diseases, obesity, diabetes, cardiovascular diseases, high blood pressure. Now just after 2 days there is a conference, a workshop actually in CFTR around high sodium in the food, you know, how one can actually reduce on that, you know. So these are actually the climate change problem. Every day actually the temperature, actually average temperature is rising, you know, and then there will be actually a problem or a difficulty to grow the food products. And that's why even in a sustainable development goal of United Nations organizations, you will see the zero hunger by 2030, we have to reduce, right? India actually ranks in 108 in a global hunger, you know, very close with the most of these African countries, very close with the Afghanistan, you see. So where this food being is going. One side we tell India is actually a food basket of the world, but food sufficiency is still the challenging, right? And then we will be actually looking for the response, consumption and the production also, so that the food security is met, you know, food sustainability is met. And then you will see, you know, in fact during the inauguration we have seen actually the wealth from the waste, how this can be actually generated, and even the first speaker of this session also we have seen how the waste like a fish waste being used for a value addition, especially with reference to recurring several of the bioactive molecules, you know. So what this actually slide is telling is that under the climate stress, food system complexity, which we are actually looking for, and then molecular sciences. So that is actually is possible through the chemistry approach, you know. And we have to look for the bioeconomy also, so that you actually are preserving the nature. You are actually ensuring that our future is sustainable, you know, and again the health oriented, you know. Now you will see the microplastics in the environment, the pesticide residues, the environmental pollutions, you know. So we are actually going to mask this effect and ensure that how our health is protected, how this development is sustainable, yeah. With reference to the food innovations, you will see the chemistry, you know, which I was referring to. As you know that food is a complex chemical matrix, right? The carbohydrate is there, protein is there, fat is there, minerals, trace elements are there, you know. So everything's again the chemistry. Then nutritional quality again is governed by the molecular compositions, right? Then you will see the several bioactive substances, which you will be analyzing by the chemical approaches, by the analytical methods. Then we look specifically from the fermentation or microbiological perspective, the fermentation. And I was just actually talking with reference to the soybean fermented products, you know. We don't actually consume much fermented product, you know. But if you go to Japan, most of these people actually consume most fermented product, whether it is a soya or whether it is a rice product or any other legumes, So you consume more fermented, you're supposed to be healthy. You will have a better healthy life and you will live longer also, So this is what actually the importance of the fermented food is. So what is this fermentation is? Actually this is a controlled biochemical transformation of the raw material into the value added product. So it actually does serve the two purposes. One, preservation, and another is the value addition. And this is nothing but the more modern food technology, I would say. So by integration in the food chemistry, we will be looking for the microbial biotechnology, how microorganisms can be used for the producing the value added product, how microorganisms can be used for conserving or preserving the food for a longer time. And then of course the nutritional science, because we have to have the nutritional security also. And as I told that hidden hunger, especially with reference to the malnutrition, with reference to the anemia, is one of these major issues actually these days being solved and has to be actually addressed. Okay. So this is what actually we are looking for a food production through the chemistry approach using a microbiological or microbial system, and we know the very simple bakery or any beverages, you give the sugar or the carbohydrate to the microorganisms, they will convert the end product by pyruvate metabolism. One of the simplest procedure which no other organisms can do or living things do, which microorganism can do. So you give them a sugar, they will convert into the pyruvic acid, and pyruvic acid by anaerobic metabolism, you get a range of the different products, right? And then you actually use that product as a functional food. So what is this actually the functional food means? Which will actually have a ability to provide the nutrition behind basic nutrition, right? In fact, I am associated with this functional food of this BioE3, and this is what actually we are looking how the functional foods derived product can be developed under the BioE3 group, Okay. So what we are actually looking for the food functionality and the nutritional quality that has been being actually governed by molecular structures. People initially were not much bothered what is the composition, what is the proximate, what is there the ratio is, but now it is more important to know, any food labeling is there, the proximate, the undesirable, the any adulterants, molecules and all these things, right? And then fermentation actually makes all this magic. Why? Because actually it biotransform into the raw material into the value added product. It biotransform into any non-palatable into the palatable one. It biotransform into the enhancing the flavors, and more importantly detoxifying also. The several of the substances like the phytic acid is there, So that actually you can do. Or the like in soya you have the off flavor, a beni flavor. You do the fermentation, you can actually reduce such a beni flavor also and make it more palatable or for the consumption purpose. Okay. Now you see microorganisms, we call it as a biochemical engines of the food production. Why? You see the several such approaches because they produce several organic acids, EPAs, then antimicrobial substances, bioactive substances, but they should be actually gross, generally regarded as a safe. So these microorganisms, which otherwise we use in the food production, which we use in the fermentations, these are the grass status microorganisms. What actually fermentation offers, as I told already, nutritional value. Just like you have suppose a milk and then curd, so you see the nutritional value, or the raw fruits and then fermented beverages, you see the nutritional value. The shelf life, right? You can actually keep the product for a longer time. Then functional properties actually you can improve, and then low ecological footprint. So this is what actually more important these days, how you can actually use a minimum carbon footprints, okay, and which I will be talking, which is actually much more comparable with the synthetic processes. So this is what actually chemistry always are being used in the industrialization, but where the drawbacks are, actually we will be actually seeing in the subsequent slide. So you see actually here, the very simple processes, we does this fermentation of this fruit vegetables, the dairy products, right? And what they do is, they actually converts, you get this fermented product, and you get the several antioxidant, the antimicrobial substances which actually can kill the pathogenic bacteria, the probiotic, anti-diabetic. You see the several health effects, you know. And this health effect comes because of what? You see, the neurotransmitters, the organic acids, then vitamins, bioactive peptides. So such several bioactive or industrially useful molecules are the functional molecules you will be getting in that fermentation processes, you know. And this actually you can do it in the industry or in the laboratory scale. So this is what the process, and you can actually accelerate the production because we know several of these biochemical pathways. So through the method of genetic engineering, it is possible to produce this product in a larger scale, in a fermenter, in a laboratory, and modulate the biosynthetic pathway by the genetic engineering process and accelerate the product in a higher amount. Okay, so all this magic actually is possible only through the microbial process, only through the fermentation technology. That is what I am trying to tell. Yes. So hence the microbial metabolic pathway that generates bioactive diverse molecules, you know. Yes, through the chemical synthesis you can do actually this magic, but the nature's diversity which you can actually tap is enormous through the microbial processes than even the chemical synthesis. So this is what actually, and this actually helps for the enhancing the functionality, then safety also. As I already told that anti-nutritional factors you can reduce, the detoxifying substances you can degrade or decompose, you know, and then you can actually consider this is a sustainable process. Why? Because we are actually not harming the nature. We are not polluting the environment. We are not actually generating any waste through these processes. Yes. So here are actually some of these microbial metabolites which can be considered as a novel food in a different food system. You know, you have this in, you have suppose the for this you have to actually explore the endogenous microbial strains which actually is required. Then you can actually do this chemical analysis, the microbial profiling, and identify such new substances. Then characterize those, you know, the several such analyses are required. And then more importantly, look for the health benefits. Now actually having the health benefit of the fermented product or any such bioactive molecule present in the food system through the preclinical and clinical trial are the actually the recent development in this field. And then you can actually look for a development of a technology or the product. So that's why actually the translation research is, you know, and ours being a translation research department in CFTRI microbiology, what we are actually looking for, developing a microbial process where the basic work is carried out and looking the scale up studies, looking the product development, looking that this product can be transferred to the industry. Okay, and what actually this translation potential actually offers functional ingredients, what are actually such molecules are present. So all this thing actually is needed. Then you see the clean label food additives, which is actually very important these days because unless and otherwise it is a clean label, we can't actually consider it as the grass or as the useful material. And then of course the sustainable nutraceuticals. So you will see such several approaches like exopolysaccharides produces there. Though it can be used as a thickening agent, but it has actually very good antimicrobial activity, immunomodulatory properties. Okay, and we actually investigated, which I will be talking about the anti-diabetic and hyperlipidemic activity of the CPS also. Okay, then you characterize those molecules and then identify such a novel or new compounds. Okay, so advanced metabolic profiling through analytical chemistry or analytical tool. And then you characterize those, and then we develop this microbial bioactive functional foods, you know, as a food ingredient. So this is what actually the important area of the research is. Why and how microbes and their metabolites are important in a few just lines I will tell, because when this microorganism which we are consuming as a food grade or food fermenting microorganisms, they actually helps to develop a stable gut microbiota in our gastrointestinal tract. Number one, that does. Second, they actually produces several metabolites that actually has a health enhancing property. What is that health enhancing property? They have immunomodulatory property, you know. So this actually is very important with reference to what I was telling, what this gut-brain axis or gut-organ axis, you know. So they actually provide this dual function. One is modulating the gut microbiota, maintaining the balance between the good and bad bacteria, and then second one is producing the several bioactive substances like short chain fatty acids, you know, or lactoglobin and then casein, and that has actually the several health beneficial properties. So I will not talk much with relation to this gut microbiota or this gut microflora, but then you see this fermented product, in other words, because of their enrich in a bioactive molecules, has, you see, lower cholesterol, you know, because now the cardiovascular diseases, cardiovascular risks are actually increasing, more numbers. Then you see the enhanced nutrient availability. You know, bioavailability is another major issue actually in the food science, right? And most of this actually the mineral deficiency which we are actually reporting, you know, is a problem because whatever through the exogenous source which we take is not sufficient or not available, is not bioavailable. Immune system boosting, you know, during COVID time we actually could understand that if a weaker immune system is what happens. So what I am trying to tell you is the fermented products are the bioactive substances present in the fermented products offers all these advantages. Okay, so that is what actually I wrote here. The food matrix stabilizes helps to provide this bacteria in a live form which are needed to maintain a good homeostasis in our gastrointestinal tract, and then this bioactive molecules helps as a functional metabolites and are associated with gut immune and metabolic axis, you know. So all these molecules has to be studied, characterized, and their functional property has to be evaluated first through the cell line, then in animal model, and then subsequently with the clinical trial. Yes. Now why we are actually looking for the microorganism as a food for our daily consumptions, because these are actually the very general information which for the students can be useful, because we will have the 50% of the habitat land is being used now for the agriculture, you know. So we have only the 50% land is now left over for our occupations. Then you see the agriculture is the primary driver for the loss of the biodiversity, you know, and then deforestation. Most of this area has been actually is being used, you know, and then there is no actually the agriculture land available additionally. That is what I am trying to tell. And then we need 80% more food by the end of this century. And from where this food is going to come, you know. And then you see, more importantly, the 1/3 of the global food system is the major cause for the climate change and emission greenhouse gases. So most of this actually the climate change is happening because of the deforestation, because of the methane emissions, because of the increase in the carbon dioxide in the environment, you know. And that's why we are actually looking, you know. And then you will see, with reference to malnutrition, the hunger and malnutrition is being increased by 2014. You know, so it is not the same what it was before, okay? And that's why we actually look for microbial food additives, you know, as a functional food. So these are actually the reasons, as I told already. You see, by 2050, we need more 56% more food, you know, because the population will be by 10 billion, you know. Already India actually is number 1 now in the population, yes. And India and China itself is half of this population of this world, along with Bangladesh and Pakistan, all right. So you see, by 2050 we will have 10 billion populations and 56% more food we need, almost double of this food, you know. And presently what we are actually using 50%, we are currently actually using the 50% agriculture land. So if we need a double of this food production, whatever we have in India today, we need 1 more copy of that, you know, then only it is possible. So from where you are going to get the additional India? You can't hire or you can't construct or you can't buy, you know.
No? So you need that much agriculture land again. Whatever the agriculture land we have, that much double of that we need to produce the food which will be useful or will be enough for the growing populations. That is what I am trying to tell. And then in addition to that, because of the industrializations, we have to actually reduce the carbon footprint, the emissions, you see. So what we have to look is improved feed, which will be with a less carbon footprint, resilient crop, the topic which was being actually discussed morning also, and plant-based burgers also, because otherwise it is meat productions. Now actually we will realize the importance of microbial system through this slide, you know. So if you have to produce the food using a microorganism, how much less you need the energy, the carbon footprint, the water. So you see very clear actually, water footprint for the microorganisms if you are producing just a 20 to 50. But for wheat, 1 kg of wheat you need 1827. And for pork 6000. You see the clear difference. Carbon footprint you see, especially for the pork, 12. You see the land how much is needed. Because this microorganism you can grow in a test tube or in a flask or in the fermenter, and then you can actually get the food product. But if you have to grow the wheat, you need the land, you need the water, you need the carbon and all these things. So this is what actually a very classical example and comparison is given among the three different systems, the animal, plant and microorganisms. So you people have to vote for a microorganism, in other words. And then you see here, the clear difference also is given with reference to the nutritional fact that if you grow the from microorganisms, you can get the protein also, carbohydrate and fat also, and then you see the proportion, compositions or the ratio, which is more desirable for our consumption. Because if you eat pork, you get more fat. If you eat wheat, you will get more protein, right? So this is what actually the difference is, and that's why microbial food, which otherwise also we are consuming, the bacteria we are consuming, the yeast, Saccharomyces we are consuming, right? And mold we are consuming, and through which form presently we are consuming through this one or the fermented product, right? So this is what actually I am trying to tell, no? And they are actually helps to produce several fermented products that enhances the flavors, the value addition of these products, all these things actually you can see here, you know, the nutritive value being used, being increased, and then digestibility, the food, digestibility of this food product, okay? And then more importantly, you will see the sensory, the flavors, and then of course the preservation also. No? So all these are actually added or being incorporated when you are actually consuming the food product which are of the microbial system. Okay? This was actually this information few years back with associated with the anemia or malnutrition problem in several of the districts in Karnataka. In the next few slides I will be finishing. Okay, which you can clearly see, and what this actually newspaper tells that several of these fermented beverages itself you can actually consume and eliminate the anemia problem. Very simple to understand. So with this aspects I will be just talking about few of these microbial probiotic product which we have developed in our lab and what are their advantages. Okay, just maybe 5 minutes. So this is one of such product, a bifidocurd. Bifidobacteria actually present or colonizes in our GI tract, especially in the colon, and a very useful microflora for a gut homeostasis as well as inducing the immune response. Okay, so this actually is technology actually we have developed, and we are actually supplementing through the milk in the form of a curd. So bifidocurd, you know, and very useful for a immunomodulations as well as we tested actually in animal model against arthritis, and you see the arthritis induced animals, you know, and then you see when we actually given this bifidocurd to the arthritis induced animal in a mice, the arthritis symptom was reduced, right? Very similar with the paroxycam treatment. So paroxycam actually is the standard drug being used commercially or in a clinical practice, and when we give our probiotic product, this was actually the difference. Just the next two more products, yeah. This is actually the shelf-stable probiotic curd which was actually tested for the diabetics, which I will be explaining, and this product actually can be stored in the room temperature more importantly, because this has actually a biopreservation property or ability to kill the spoilage microorganisms. Okay, and then you can actually see here that zone of inhibition which we are seeing here because of the antimicrobial compound produced against the pathogenic bacteria, just in the 2 slides. So what we did actually, we tested for this in the animal model, what is actually the immune response as well as the disease prevention ability, and then you can actually see that it helps to reduce the pathogenic microbial load and increases the useful microflora. And this product actually we checked even in the clinical setting, and then you can actually see increases the HDL, reduces the LDL, and then especially the pro-inflammatory cytokines. Okay, so this is actually has a very useful health promoting ability with reference to the immunomodulation as well as the gut microbiota modulations. The last product. Okay. Yes. So summary of those products actually is described here. Here actually we incorporated the 3 bacteria. One is the bifidobacteria, another is the lactobacillus, and then third one is the lactococcus, because lactobacillus actually colonizes in the smaller intestine. And then actually we have actually this product also can be stored actually at least till the room temperature for a week in a room temperature, and is described here with the storage. Okay, and what we actually did is, you see here, we checked for the immunomodulation properties, and this actually can decrease the pro-inflammatory cytokines in the animal models and induces the anti-inflammatory cytokines beside the gut microbiota modulations. The last one actually I will be explaining with reference to the, so these 3 products were the live bacteria which we have incorporated. Now here actually we use the cell-free extract of the probiotic bacteria which is actually called as a postbiotic effect. And here actually we are looking for the alpha amylase, alpha glucosidase, and pancreatic lipase inhibitions. So very clear actually results are there. You see, the 2 different concentration of that culture filtrate inhibit the alpha amylase and the alpha glucosidase, and that's why subsequently what did we do is we checked for the anti-diabetic activity, in the animal model, and we actually could see through the histopathological that was just like the conventional drug, even this molecule act for a prevention of this diabetics. Okay, I know time is little bit short to explain all these results. This is of course the DRDO's project which we are looking for. So with this, we are actually promoting some of this product through this organization like this MBSI. There is a buttermilk, and then this actually we are preparing using the bifidobacteria. The story about the bifidobacteria I will not be able to tell now because of the short of the time, So just a few information is here on that, and then I will be actually explaining the with relation to the sustainable development, and then just for a 30 seconds. Okay, so with relation to the sustainable development, definitely this information is very important, which I have actually tried to bring it, that how the sustainable development and then sustainable society actually can be explained in advance and in a much better way. How these green revolutions and then other revolutions actually has helped us to achieve the sustainable development goal. Next slide is more familiar to most of these people. Okay, the Karnataka map, and if you invert this Karnataka map, you see Japan map. I hope I could stabilize my 30 seconds, and then this pictorial representation, I think, picture, and subsequently even this also. So this is actually the really interested with the what I have written there, that marching towards sustainable society. So how you are going to link this bridging the chemistry and food toward the sustainable society is actually the answer which I am trying to present here. So with this 2 very simple bullet points, I am concluding this my presentation with reference to the microbial metabolite, their health benefits, and how actually they actually help us to maintain a sustainable society, in other words. So with this, I am just quickly almost about to tell you everyone thank you very much with this presentation. I will be very happy if you have any questions, or you can actually, of course, write to me,
Thank you
for further interactions.
Prakash, for the nice presentation, which was very informative, and within a short period, I think it will give so many information about the sustainability of food. Thank you. Now the floor is open for questions. If any, we can take 1 or 2 questions quickly.
Very nice presentation. And my queries are like microbial metabolites which you have, they will be synthesized by synthetic pathway. And by the chemical synthesis, we are controlled by reaction mechanisms.
Yes.
So whether the same level of a control or a kind of any control of the microbial metabolites is possible or not?
Yeah, through the chemical synthesis also it is passed just like lactic acid, right, or acetic acid or vinegar also, which we are actually developing. But then the environmental pollution, the carbon footprint, the energy requirement, and then the diversity you will not be able to achieve, which is possible through the microbial processes. Yes.
Any questions from the students? If no questions, I think we will conclude this session, and I
thank you,
Dr. Prakash, for maintaining his time and for the nice presentation. Thank you.
Sir, excuse me. Sir, I request you to please be seated.
Oh, still this.
Thank you.
I thank Dr. Prakash M. Halami, sir, for presenting his talk on microbial metabolites,
A novel food for sustainable society. Thank you, sir. BM, sir, for chairing this session. Thank you, sir. Now we'll have a lunch break for about 1 hour. I request all of you to please gather back at 2:30. Thank you.